The vast majority of life encompasses a variety of complex organisms with distinct functions and features. In biology class, we studied the functions of these organisms and how they interact with their surroundings. Now it is time to apply these concepts to the real world.
Placozoan are small, three-layered organisms that have the smallest DNA. In this study, researchers focused on specific species of placozoan known as the Trichoplax H2. Through sensitive DNA and RNA extractions, placozoan were sequenced based on similar microbial communities.
Microbial communities are groups of organisms that share a common living space. They interact with each other, often through competition or symbiosis. The purpose of this investigation is to study the symbiotic relationship between bacteria and the host, Trichoplax cells.
As thousands of host individuals of the Trichoplax species were analyzed, scientists discovered a pattern in the symbiosis function at the cellular level. Phylogenetic analysis placed the Trichoplax with the bacterial family Midichloriaceae, also known as Rickettsilus. The second consistently present bacterial species was the Marglus bacteria, also known as R. illudens.
Figure 1 in the article represents a phylogenetic analysis of the Trichoplax H2 and the two bacterial species. There is a close association between the G incantans phylotype, which is placed in the Rickettsial group in the Trichoplax H2. R. illudens is also placed in the category of Margulae bacteria.
Fluorescence in situ hybridization, also known as FISH, is a technique that linked bacterial sequences to their distributions in the Trichoplax. Figure 2b describes the intracellular nature of the bacterial symbionts and the spatial segregation. Figure 3 represents the results of the tomographic reconstructions, which shows how G incantin is embedded within the rough ER of the cell.
R. illudens undergoes a complete TCA cycle and generates energy and biomass from glycerol and the oxidation of fatty acids. The source of glycerol and fatty acids is most likely the algal diet of the host.
In class, we studied fat metabolism in a general context. Glycerol is converted to an intermediate of glycolysis, and the fatty acids are oxidized to acetyl-CoA. The acetyl-CoA is then further oxidized in the citric or TCA cycle.
Figure 4 illustrates the primary biosynthesis pathways for both the R. illudens bacteria and the G incantans. Lipase from the host cell hydrolyzes lipids to glycerol and fatty acids. The fatty acids are catabolized to acetyl-CoA and respired in the process of the TCA cycle and beta oxidation.
While R. illudens primarily uses beta oxidation of fatty acids, G incantin depends on the import of nutrients from the host cell. Although G incantin primarily has genes for energy and parasitism, it is still able to synthesize ATP by itself. Also, the total number of G incantin cells per host is roughly the same as the number of host cells. These factors lower the detrimental impact on the host cell.
The last important concept is the transmission of the Trichoplax symbionts to offsprings. As we studied in class, evolution requires heritability and variance within a population. In this case, however, the host individual follows a mitotic process to pass bacteria during cell division. This leads to an invariant trait across this phylum that affects future generations.
In studying organismal interactions, it is very important to understand and apply the concepts to a practical field outside the classroom. This research combines the various concepts of evolution, ecology, and metabolism, as well as genetics, to provide an account of the functions of organisms in the ecosystem.